Section 6 of The Psychic Life of Micro-Organisms by Alfred Binet. This is a LibriVox recording. All LibriVox recordings are the public domain. For more information or volunteer, please visit LibriVox.org. Recorded by Leon Harvey. Chapter 6 Fecundation. We now enter upon a subject fraught with obscurity. We shall limit our investigations to ciliated infusoria, as it is among these species that fecundation and the psychical phenomena attendant thereon have been observed. Ehrenberg had established by his authority the prevailing opinion in science that copulation never takes place among infusoria, and that all facts observed by early writers as connected therewith are to be regarded as phenomena of longitudinal fissiparity. This erroneous idea prevailed unquestioned until 1858, when M. Balibiani addressed a communication to the Academy of Sciences, wherein he showed that sexual reproduction, preceded by copulation, is found among infusoria. Before entering upon a description of the changes that take place in the nucleus and nucleoli of infusoria in question, we shall briefly sketch the course of psychical phenomena through which the ciliated infusoria pass when making ready for copulation. We shall follow in the footsteps of M. Balibiani, freely using his descriptions, the exactitude of which has since been confirmed by Gruber. To appreciate fully the significance of the facts to be adduced herewith, we must recall to mind that through the entire animal kingdom, the act of sexual coition is invariably preceded by an introductory manifestation of psychical activity, which may last for quite an extended length of time. The female, when pursued by the male, seems to be animated by two conflicting desires, that of yielding to the male and that of repelling his approaches. This show of unwillingness, which is but temporary and more seeming than real, has the effect of inciting the male to attempt an exhibition of powers calculated to capture the female. According to M. Espines, who has thoroughly studied this subject, there are 5 classes of phenomena which assist in preparing the way for sexual union. Firstly, provocative contact, the lowest of all these phenomena. That is the one which most approximates to the physiological order. Secondly, odor. Thirdly, color and form. Fourthly, noise and sound. Fifthly, play or other variety of movement. It appears to us that almost all manifestations of love in human beings themselves could be classified into these 5 categories. Among the simplest forms of life we meet with incipient traces of such aesthetical manifestations pointing towards the preparation of 2 animals for sexual intercourse. It is curious, remarks M. Balibiani, to find among these organisms, which all zoologists, by reason of their diminutive size and extreme simplicity of structure, have placed at the remotest limit of the animal kingdom, acts that mark the existence of phenomena analogous to those by which the sexual instinct is exhibited in a large number of metazoans. Upon the approach of the period for propagation, the paramecia come in from all points of the fluid and assemble like little whitish clouds in more or less numerous groups about the objects that float upon the surface of the water, or adhere to the side of the vessel containing the tiny artificial sea which the animalcula held captive. Intense excitement, which the need of food does not suffice to explain, prevails in each of these groups. A higher instinct appears to dominate all these tiny organisms. They seek each other's company, chase each other about, feel here and there with their cilia, adhere for a moment or so in an attitude of sexual coercion, and then retire, soon to begin anew. When these minute assemblages are dispersing by shaking the liquid, they quickly form again at other points. These singular antics wherewith animalculae appear to incite each other mutually in copulation often continue for several days before the latter act is definitely effected. Other infusoria, particularly the spirostomes, seek the deep spots of the liquid, or bury themselves in the oozy sediment of the bottom, not to come forth again until they have separated. The stentours have different habits. They are fixed by their pedicles to submerged vegetable patches, which they often come like small, closely mown lawns, of a green, brown, or blue colour according to the species. They turn the forward part of their bodies, which is elongated in the shape of a trumpet, about in all directions, and seek to unite with each other by the broadened extremity which corresponds to the bell of the trumpet. Among the numerous species forming part of the group of Oxytrichinae, The active coition likewise exhibits certain interesting preliminaries. The two individuals whose bodies are generally very much flattened, and of which the lower sides are provided with cilia at times strongly developed, superpose themselves upon each other on the ventral side and mutually entangle the cilia which cover that region, while with their cornicles or anterior tentacles they touch repeatedly the different parts of each other's bodies. These introductory moves frequently last for several hours before copulation begins. As regards the act of copulation itself, it is of exceeding interest to the psychologist who can admire the precision with which the two individuals assume the attitude necessary for fecundation. During conjugation, the two ciliated infusoria are always joined together at the aperture which forms the mouth. It has been thought that this aperture must play the part of a sexual orifice through which the two animalculae in copulation effect the exchange of reproductive matter. It has been suggested, moreover, that the especial sexual orifice was present, quite close to the mouth, but these questions of structure are still doubtful. The attitude of these organisms during copulation varies according to the position of the mouth, which in certain groups is lateral and in others terminal. The greater number of species have a lateral mouth. To this class belong the Paramycia. These infusoria, in which the buccal fossa lies at the bottom of a deep excavation made in the ventral face, cover each other over the whole extent of this face, excluding a glutinous substance which causes them to adhere to this position. The two mouths then lie exactly upon each other. Copulation lasts from 24 to 36 hours with the Paramycium aurelia. It lasts several days, 5 or 6, with the Paramycium bursaria. Among the Oxytrachinae, the two animals in conjugation blend together as an important part of their persons in a very intimate fashion. We next arrive to the second group of infusoria, which show a terminal mouth. Of this type we have a specimen in the Didinium nasutum, the curious hunter infusori. We may further mention the Colleps, the Nasula, the Prorodon. The two organisms in this case do not embrace laterally. They take a position end to end, connected by their anterior extremities, mouth opposite to mouth. Then little by little, while still joined at the buccal extremity, they shift about until they meet length to length. We shall mention particularly but briefly the curious phenomena that accompany fecundation among the vorticells. Even more than the instances just cited do these phenomena resemble the process of fecundation in higher animals. For in this instance, fecundation is effected between two differentiated individuals. One of each acts as a male element, and the other as a female element. The vorticells are colonies of infusoria, which are found sedentary individuals, having the shape of minute jugs, and also detached individuals called Micro-Organizms, which are formed by repeated divisions upon the colonial tree. These Micro-Organizms have exactly the same mode of locomotion as the spermatozoids. Engelmann has followed their movements. He is seen then swimming about, turning upon their axis for 5 or 6 minutes. Then, having come into the vicinity of a vorticell, they abruptly change their manner of movement, capering about the latter like a butterfly flitting about a flower, touching it, retreating, and then approaching it again and apparently feeling of it. At last, after having visited the others near by, they return to the first one and fasten themselves upon its surface. The coition is not effected without a certain show of resistance on the part of the vorticell. It hastily contracts the peduncle to which it is attached, at every touch of the micro-gordidium, while the latter, in order to prevent itself from being thrown back by these rapid shocks, and in order to be always close to the individual with which it wishes to unite, fastens itself by an extremely fine filament to the style of the vorticell. Thus attached and drawn along with the movements of the latter, it finally succeeds in effecting a junction with it, and in penetrating into its body. It is now time to describe the material phenomena that takes place in the interior of the two infusoria, and which constitute the material act of fecundation. The psychical manifestations which we have just noted, and which so strikingly resemble the manifestations accompanying the copulative act in higher animals, are of themselves sufficient evidence that this conjugation is a sexual union. The material changes effected inside the bodies of infusoria in copulation do not extend to all their organs. The main mass of the body, the protoplasm, plays but a secondary role in the matter. The change appears to be effected exclusively in the nucleus and the nucleole. Let us further state that, So far as is known, these changes are never effected apart from coition and before the infusoria actually embrace. Copulation sets in every time, apparently, that these animals, under particularly favourable conditions, have actively reproduced by fission. Fissiparity is then seen to cease and conjugation appears. We have not the time to sketch the history of this important question of physiology, interesting as it may be. It will be enough to recapitulate what we actually know upon the subject, taking as our guide substantially the views of M. Balbiani, who, as is known, was the first scientist to study the psychical phenomena connected with the fecundation among infusoria. The divergencies between his observations and those of another eminent investigator, M. Bütschli, extend in reliability only to points of detail. Let us first mark the modifications that take place with the Ilona Gheorghiesh during conjugation. Each of the two infusoria in copulation possesses a nucleus, endoplast, main nucleus, and close because this nucleus, an organ considerably smaller, a nucleole or attendant nucleus, or a latent nucleus, endoplastule and accessory nucleus. This minute body must not be mistaken for the nucleole that is often found in the interior of the nucleus among many microorganisms and in cellules. It has a function entirely different. Of these two elements, the nucleus plays an almost negative part in the act of fecundation. It assumes irregular outlines and becomes rumpled, while its contents collect in detached masses of various sizes. It grows clear by degrees and is finally absorbed. It disappears, accordingly, by a phenomenon of regression and without dividing. Fecundation aims to replace this wasted element by a nucleus of fresh formation. The latter is produced at the cost of the little body we have described by the name of attendant nucleus or latent nucleus. The attendant nucleus does not act in making up a main nucleus, in the cellule of which it is a part. It finds its way into the body of the other animal, and it is in this new cellule that it is destined to perform the function of a nucleus. In the Chelidon cucuruleus, the attendant nucleus divides into two striated capsules, never more. These two capsules grow to unequal sizes. The largest attains the size of 40,000ths of a millimeter. Is this one that forms the new nucleus of the Chelidon. The second capsule shrinks and becomes compressed. It takes its place beside the first one and constitutes the new attendant nucleus. To the study of this type of fecundation, we may limit our attention. It is the simplest of all, and other forms may be comprehended within it without much difficulty. What complicates the process in the other species is principally the successive modifications through which the old nucleus passes before suffering absorption. In the Stentor coeruleus, the nucleus has the shape of a long chaplet, or a string of beads. At the moment of fecundation, the beads of the chaplet break apart and spread in the protoplasm, where they finally become absorbed. Among the Paramecia, the phenomenon is still different. The nucleus, at first massed together in a cluster, lengthens out into a very long string which breaks, and the pieces becoming scattered about in the protoplasm are absorbed. We find that fecundation in every instance introduces the dispersion and disappearance of the old nucleus, and that the latter is replaced by a new nucleus resulting from the transformation of the attendant nucleus that proceeded from the other organism. The various modifications presented by this attendant nucleus likewise contribute in great measure to the complexity of the phenomenon. We have seen that in the Chelidon the attendant nucleus breaks into two globules, of which one goes to form the new nucleus and the other the new attendant nucleus. Matters take a different course in the Paramecia. In the Paramecium bursaria, for instance, the attendant nucleus divides into two and then into four capsules. One of these capsules suffers absorption, a second one becomes the attendant nucleus, and the two others coalesce with what remains of the old nucleus to form the nucleus proper. In the Paramecium aurelia, the division is made into 8 capsules. 3 are cast out, and of the 5 left, 4 are meant to form the new main nucleus. In reality, each paramecium segmentates first into 2 and then into 4 divisions, and each one of these 4 individuals takes one of the capsules. The 5th capsule is designed to form the attendant nucleuses of these 4 organisms. It divides accordingly into 2 and then into 4 parts. That is to say, into as many parts as the body of the animal divided. There is no question in our mind but that conjugation in this case is a sexual phenomenon. A circumstance that at the outset confirms this is the peculiar maneuvering the animalcula go through before abandoning themselves into copulation. The movements they execute admit of exact comparison with the actions attendant upon copulation among higher animals, but we shall recur further on to the physiological significance of conjugation, when we shall endeavour to explain, according to the most recent investigations, the function of the nucleus in the cellule. The question may be asked, what is the starting point, the provocative of these sexual phenomena, the cause that sets them in play? Butchley justly thinks that conjugation is determined by internal causes. In fact, it takes place directly after very active periods of spontaneous division, as Binet has shown. When we bear in mind that the object of conjugation is to replace the old nucleus which has become wasted and worn out, we may conjecture with some degree of likelihood that the physiological condition of the nucleus constitutes the sexual excitant that causes the infusoria to copulate. However that may be, a curious observation within the paramecium aurelia has made us acquainted with one of the structural conditions of the sexual instinct in that infusory. For a long time, J. Müller has pointed to the presence of filaments in the nucleus and even nucleolus of paramecium that had the appearance of spermatozoids. Observations to the same effect have increased since then, and it is now known that the filaments are schizomastocytes, parasitic bacilli, which find their way into the nucleus and nucleolus and multiply, after their customary mode of segmentation, by disarticulation. Balbiani has definitely determined the nature of these filaments by morphological and microchemical methods. He has found out, among other things, that the filaments do not dissolve in strongly concentrated alkaline solutions, and has known that bacteria exhibit this peculiar attribute of offering a great resistance to destructive agents. In the nucleus which they have penetrated, these parasites induce a pathological condition that results in destroying every manifestation of the sexual instinct in the infusory. Among a swarm of animals of this species are that in copulation. Single individuals are found that show a nucleus and nucleolus completely charged with bacteria. Sometimes these organs suffer an enormous dilation, the nucleus becoming nothing more than the enveloping membrane, which is filled like a huge pouch with parasites. The animal continues to live, but it no longer attempts to copulate. End of section 6.